Good morning, everybody. My name is Ian Cotgreave, and I will be guiding you through this webinar for the morning with my colleagues. And the webinar deals with work going on within the Mistress SafeChem program, within a work package dealing with hazard and exposure screening tools for Safe by Design. And we have a few rules to follow during this webinar. Please put your questions in the chat. And we'll deal with them at the end when we have time to deal with those. Keep muted while you are asked to unmute, until you're asked to unmute. Turn your camera off, please. The presentations won't be sent out, and the webinar will be recorded and available via link, which you'll get in an email. And the presenters here are shown in the next slide. Hazard and exposure screening tools for Safe by Design chemistry. Once again, hammering home that these are the rules and regulations, so be aware of them. So we'll keep order. So we keep the questions to the end, but write them into the chat, please. Of course, the Mistress SafeChem program, generally describing this, it's a 4+ hopefully 4-year program, or 8-year program in total, 2-stage rocket, and its purpose is to apply new approaches to designing chemical process and products with a Safe by Design angle, and where we introduce risk assessment and life cycle management early on in process modification, process changing, and so the whole thing is aimed at sustainable chemistry and process in the future in Sweden as a platform, developing a platform. And these are the partners you can see in the program. They're both universities, institutes, and companies are involved here. We have a number of other interesting partners here as well, particularly ICAM, the industry organisation, and also the NGO. NGOs are represented here as well. This is our website, mistressafechem.se. This is the Mistress SafeChem vision to enable and promote the expansion of safe and sustainable and green chemistry industry in Sweden. It's a tall order, but we are attempting to approach this from a safe science perspective. And these are the speakers for the day. So Ulf Norinder will start talking about in silico hazard and screening toolbox which we've developed. I'll take over and talk about some in vitro cell biological screening tools that were also available within the program. Then we'll talk, Ulrika Nilsson from Stockholm University will talk about the chemical screening toolbox. Of course, hazard is one thing, but we need to know the exposures as well to develop an LCA and risk assessment approach. And then I'll deal with some thoughts around hazard and risk screening and its coupling to LCA in the program. And so, without further ado, I'd like to give the word to Ulf Norinder. Can somebody please mute? I'm hearing a lot of things in the background. Thank you. So Ulf Norinder from Stockholm University Department of Computer and System Science. Can you take the word, please?
Yes, I will. So welcome to this breakfast seminar. I will talk about the in silico hazard screening toolbox that we started to develop in the project. So next slide, please. And I also want to mention here how it sort of connects to the green and sustainable chemistry, the principles there, key components. And I hope that I will be able to convince you that we do influence the design and sustainability and recycling, and also work on efficient and safe syntheses, as well as chemical building blocks, and hopefully also at the end that influences safer industrial and consumer products. Next slide, please. So when you develop a hazard screening toolbox, you have a whole bunch of data sets, that is experimental data and chemical compounds that you like to build some sort of mathematical model for, where you relate the properties of the structures, which you see at the bottom left, described in a language called SMILES, which you see at the very bottom. And if you read enough of these, you can actually visualize the structure more or less as a computational chemist. With these data sets and the experimental data, we try to describe the structures with what's called descriptors in various forms, which could be molecular weight and lipophilicity and electronic charges and things like that. And then you try to develop some sort of mathematical model with some sort of algorithm. And today, as you probably all heard about, artificial intelligence, machine learning, deep learning, this is what we utilize as well.
Yes.
And with that particular algorithm, you hopefully are able to develop a model that you may validate to see how it performs and also determines its applicability domain. And I'll come back to that. So at the bottom, you see three typical algorithms that's been used over the years, and to the left you see what's called a decision tree, where at the end of that tree, at the leaves, you'll get a prediction whether the substance is toxic or non-toxic or active against the receptor or not. And then you build many of these trees, and with many trees you have a forest, and the method is called random forest. And it's quite popular. The random, it's because there's an element of randomness in when you do this. In the middle, you have something called a support vector machine, where you try to divide your two categories, solid and open circles, so that you can have a difference between them. Although in your original descriptor space, you can't see that, but this is through a transformation which you need to find. And at the right is what's a very simple version of the very popular artificial intelligence and deep learning, which is become quite popular these days. Okay, next slide. And so when you have a new data set, you calculate same descriptors, you apply your model, and you get a set of predictions from each of these models. And here it's very important that you have an appreciation of the applicability domain as well, because all these modeling is empirical, which means that the model has a limited sort of domain of where it can actually predict things with some sort of reliability, and the user needs to be made aware of that fact, whether the model can predict something with enough accuracy or not, because it's like it's easy. You can develop a model for predicting the weather the weather in Scandinavia, but sure enough, somebody's going to ask for the weather in Tokyo, and then the model is going to give you a number, but you need to take that number with a huge grain of salt, and the user needs to be aware of the fact that is way outside for what the model is actually designed for. And the reason is that bad models should get bad reputation for the right reasons, namely that they are bad models and not that people are trying to predict things way outside. Next slide, please. And so with these models, we try to predict properties and help other work packages like in life cycle assessment or in the Mr. Sapien work packages and case studies. We also predict these properties so that chemical substitution can be made. If you have a large set of compounds, maybe some of them are less prone to have liabilities, and you may substitute them that way. And for compound profiling in general to see how the toxicity and biological activity profile looks like. Next slide, please. And to date, so we developed a whole set of models. We have quite some endocrine disruptor models, for endpoints like estrogen, androgen, and so forth. But we also have a battery of toxicity models for reproductive toxicity, carcinogenicity, mutagenicity. We addressed what is, of course, very important, biodegradation of the compounds, and as well as bioconcentration, in this case in fish, but also persistence, which of course is of interest when you're developing and trying to substitute components and materials. And we are currently working on additional biological endpoints, ecotoxicological endpoints, some more models for bioaccumulation, and also various physical chemical properties important for life cycle assessment, like vapor pressure, octanomal water partition coefficient, and some more. Next slide, please. And when you do the prediction from the current model, you will get a spreadsheet looking a bit like this, where you have each of the compounds, and then you have a whole bunch of columns, basically with predictions. and you will have, whether it's active or inactive against that endpoint designated by 1 or 0. But you can also, as you see, something called inconclusive, which means that the model cannot tell whether it is active or inactive, but it's within the domain of the model, so the model can actually say something, but you don't have enough information at this point. So you need to, further develop your model to be able to resolve these inconclusive results. But you can also get out of domain, which means that the model is telling you this is way outside for and very dissimilar from the chemical structures that the model is developed on. So I can't really give you a prediction on this. Next slide, please. And basically, to conclude, I just want to mention what I started with, our connection to the green chemical principles. And I think we are, designing a less hazardous chemical synthesis, and we're doing that by fingerprinting the starting materials and make sure that if it's possible, we start with less liable starting materials with respect to toxicity or endocrine disruption or whatever. And this will hopefully we will be able to design, safer chemicals that way, which we can also profile to see whether some of them seem to have a better profile than others for whatever, substitution is. And then hopefully also we, by the use of these models, biodegradation and persistence, we can have somewhat of a handle on how these chemical products will degrade after use, and so that they will hopefully become innocuous substances and will not accumulate in the environment. And next slide, which is, I think now, this is my end. So
that's the end, Ulf. Thank you very much for the presentation. And so I'll take over now and talk about the use of biological systems in a toolbox that we've developed, and these are the authors below who are part of the project who've contributed to this, as you can see. Christina Fant, Anders Kalin, Oskar Karlsson, and Magnus Breitholtz, Mikael Mogermark, and you can see their various affiliations there. So this is a multi-faceted toolbox that we've developed here. And the word here is fit for purpose, because you can go to extremes with respect to in vitro model systems in new assessment model paradigms of risk assessment based on non-animal approaches. But we've chosen to go to a fit for purpose toolbox, and I'll come to that in a second. And we are mainly supporting decision making in relative hazard assessment where necessary, where data gaps need to be filled from the in silico work. So, and of course, at the end of the day, what we're trying to capture are specific events in known toxicities or toxicity pathways, or they're termed adverse outcome pathways by the OECD. And there is a wiki here that you can go to start to understand how the OECD has collected mechanistic toxicological information underneath mechanisms of toxicity. So these cell models are designed to capture specific and quantify specific events at either a molecular level here, as a chemical interacts with the cell, or an organella level, cellular level, and higher orders of organization as well at tissue levels. But we are making a toolbox here that's fit for purpose. And just to show you the concept of adverse outcome pathways and how cell models are designed to capture important events, this is one of them. This is an AOP on skin sensitization, which obviously is very relevant to some of the toxicities that chemical process initiate in the human. And a cell system called the H-CLAT system. It captures actually one of the very key events here, activation of cells in the skin called dendritic cells, which are immune competent cells, and they cause activation of local T cells, and you get your skin sensitization process. So the idea is to capture a specific key event, or a K3 in this case, of the AOP in skin sensitization. And so all of the systems that we've developed and put into the toolbox are related to this kind of approach. As a contrast, there are more, rather than the mini approach, we've got the Rolls-Royce approaches out in the European network of research. This is one of the larger, rather larger programs that I've been part of, the EU Tox Risk program, which is developed to provide cellular systems and computational systems to actually totally replace animals in risk prediction. And in this, there are tiered approaches for cellular. This is just one of them for the liver, for instance. When you're dealing with liver toxicity, there are tiered systems going in with increased complexity, moving from simple cell systems all the way up to precision cut human tissues. And of course, this is the Rolls-Royce approach, and this was a huge program, 30 million euro flagship program, which has just been replaced by another one. This is not the approach we've chosen in SafeChem. We've more chosen a tailored toolbox to capture some of the major hazards that one's interested in chemical design and process. So here are some of the cellular systems and their relevance to endpoints in toxicity. We have toxicity models that capture general cytotoxicity. We have a cell model that captures skin sensitization potential, phototoxicity, which is also an extremely important endpoint, particularly in chemical process where chemicals can become activated by absorption of UV light and become, for instance, DNA reactive. Genotoxicity, of course, very important, always a potential hazard at the bottom in unknown chemistry and changes in chemistry. Skin irritation, which also, again, from the work environment perspective in a chemical process development is also important. Here there are a number of tests, particularly this one here using human tissue, actually reconstructed human tissue. We also have access to some interesting test models for specific endocrine disruption potential. These are luciferase reporter assays, which can light up if you have estrogenic or androgenic potential. And we also have some pretty high content biological assays for hepatotoxicity in humans, as well as the potential for neurotoxicity. So these are the sorts of no-nos that you really want to be able to screen simply and effectively and in a tiered approach to hazard and risk assessment. And just to illustrate some of these test systems, this is the Calyx system that we'll have access to, where a chemical moves into the cell, it binds to a component within the cell, which then moves into the nucleus, binds to a piece of DNA, which actually carries and activates a little luciferase protein, and that luciferase protein is fed the substrate, which produces light. So the more light you get, the more of activation of the androgen or the estrogen receptor you've achieved, and you can quantitate this. So this is a pretty neat system for picking up that kind of potential. We also have some high content imaging assays. This is an assay platform where so-called cell painting is used to show you the morphology of cells. Oftentimes cells will tell you how they're feeling by the way they're looking. And this is called cell painting, and we can put in several dyes at the same time and paint cells up and look at various components in the cell like mitochondria, the cytoskeleton, and so on and so forth, and make measurements by image analysis. And this is automated. So this kind of approach is really quick and effective, and also can be multiplexed in quite high throughput when necessary. This is just another example of the use of these kinds of imaging techniques where we're trying to look at the potential effect of chemicals on the growth of neural stem cells and their differentiation into various kinds of important cells that you would find in the brain. And if any compounds will disturb any of these processes, they can be picked up by visualization here on instrumentation that actually quantifies automatically. And so that's another example of imaging that's used here is also from Stockholm University or SciLife Lab, is the actual in the area of ecotoxicology, where we are focused on imaging of a signal organism called Daphnia magna, which is a commonly used organism for signaling ecotoxicological potential. And in this method, the Daphnia are exposed, they are then imaged, and they can report out various kinds of endpoints by imaging, again in the same way as the cell painting experimentation, to show specific effects on Daphnia's health and viability. So this is all screening as well, and these are the guys involved in these processes. So in summary, the tools are designed to be employed as a second tier after in silico screening, when the primary hazards, and also when primary hazards covered by the test are suspected. Many a time you will know from structural evaluation from in silico that you will have a suspected, let's say, neurotoxicological potential, but there is no data out there on the structure, so you might have to provide that from your own experimentation. and you do have a model that can pick that up. So, and the tools are only employed when data gaps arise or when biological confirmation of in silico prediction is deemed important for decision making and hazard ranking. So when you're trying to compare apples and pears in terms of hazard, you may have to back up the in silico predictions with specific in vitro assays. These assays, many of them also allow combinatorial exposures, and so equipment is available for extremely small volume combinatorial exposures, which is important. And the data derived are usable, as are the in silico data in LCA analysis, in predictions presented in LCA. And I just want to point out the final thing, and I missed it on the slide when I was on it, is that these assays, most of them are OECD guideline studies. In other words, they are recognized internationally and performed according to OECD guidelines, which makes the data also interesting from a risk assessment perspective and potential submission to regulation. So we try to do that. So that's the in silico toolboxes on hazard science. And now we'll move over to the exposure. Is somebody wanted to say something there?
Ian, this is Ragnhild is my name. I am a communicator. We have a question in the chat.
Okay.
From Magnus Einival. I think it's good to take that now. Can you see?
Magnus. No, I can't see that. I'll have to drop out of the.
I can just say it perhaps. Do you hear me?
Yes, I see it. Hey, Magnus. Hi.
Yeah, hi. How are you doing? I'm just thinking, you said that this is a fit for purpose approach for your hazard screening assays. I just thought, do you have any sort of set criteria for how do you determine that in terms of how do you know that it's actually fit for purpose? I know many of these assays, I agree with you that they're really good, but I thought about this a lot, how to really know that you're choosing the right assays for your purpose, so to speak.
Yeah, well, you know, we can't. I haven't got a Rolls Royce here, so we have to be rational, and we've chosen assays that we know relate to key events in adverse outcome pathways, and we know they have OECD guideline associated to them. Right. And there's only one set of assays that really isn't, and that's the one provided by AstraZeneca. But they have enough in-house high content biology validation data to assure that their assay, the high content assay on liver toxicity, is really fit for purpose. So I agree, it's difficult. You have to make choices and you have to make a rational decision. This is the toolbox we have, it's a good question, Magnus. I do agree. Okay, thank you very much, and we'll now move on to the exposure side. So I'd like to invite Ulrika Nilsson from MMK at Stockholm University to discuss the exposure side.
Thank you, Ian, and good morning, everyone. So I want to describe, as Ian says, the analytical tools we have within MISTRA for screening of chemicals in different materials. And in my group, we are especially interested in chemicals in textiles, because we have seen that we have in normal clothes, skin, clothes, clothing, and so on, we have usually a lot of chemicals. And that might be problematic because they can migrate from the textiles and they can be taken up by human skin. So basically what we do is a very simple sample preparation. What we do is that we cut the clothes in small pieces and just solvent extract. And then we filtrate the extract and inject them into our analytical instrumentations. And the simple sample preparation has purpose because we want to still have all possible chemicals, as many of them as possible, so we don't miss a large part of compounds with certain properties. And then we have liquid chromatography, mass spectrometry, LCMS, which is for less volatile chemicals. And we also have gas chromatography, MS, GCMS, for more volatile and semi-volatile chemicals. Mass spectrometry is very powerful because it can give us mass, so the molecular weight of a compound, but also structure. So that is important for the identification. Next slide, please. So this slide is for you because I realize that many of you probably don't have a chemical background. So this is a very simple explanation what is chromatography. So in this case is shown liquid chromatography, so LC with MS detection. So chromatography means that you have a separation column and chemical compounds, they are separated on this column depending on properties. So for instance, log P, usually log P, which is in a simple way, you can describe it as lipophilicity or fat solubility. So through this LC separation column, the solvent is pumped, and into the solvent stream, the sample is injected. And in our case, textile extracts that might contain several hundreds of compounds, they are separated one by one on this LC separation column and detected after different times depending on the properties, and that is called retention times. They are detected by the mass spectrometer, MS in this case. The compounds are then coming out as you see on this screen. So the one peak to the left is the compound coming out that passes fastest through come out the shortest retention time. And then the mass spectrometer ionizes each compound. So each peak here is a compound that is ionized and broken into pieces of ions. And that gives a mass spectrum that is specific for each compound. So you can regard this as a fingerprint of a compound, but also for unknowns, it helps you to identify the compound. Next slide, please. So on the following slides, I will describe what we call non-target and suspect screening with, in this case, I chose the example with LC, HRMS, and HRMS means a high resolution mass spectrometry. HRMS is especially powerful because it gives you accurate mass, so you get a number of suggested molecular formulas. And then why choosing non-target and suspect screening? And non-target means that we search for everything basically. So a priori unknowns in textiles and other materials. And suspect screening is good because we know already that some compound classes exist in clothes. And therefore we don't want to miss that. So we tell the mass spectrometer also to search for compounds that we know should be there or often exist in textiles. So non-target means then that we tell the mass spectrometer to ionize the most intense peaks from the liquid chromatography and give us mass spectra. And suspect screening, that we tell the mass spectrometer which a list, we have a short list, we have a list of compounds that we have selected that we want the mass spectrometer to give us mass spectra for. Next slide, please. So this screening, non-target and suspect screening, all together we combine and we end up with sometimes 1,000 different so-called features. And I call them features because not all of them will be real compounds. A great part is just noise, and we are not able to identify everything. And because of the huge amount of data, we need to filter this somehow. And that we do with some requirements. So for a feature to be a real compound, it must be present in duplicate samples. So we inject the same textile samples several times. And also the peaks from the LC must be at least 5 times the peak intensity of a blank. And with a blank, we mean injection of pure solvent. And then a suggested compound must also match with a reference spectrum, mass spectrum, so at least 80% similarity. And also for a suggested molecular formula, we must have more than 500 literature references. Next slide, please. Then we have added an extra filtering step, and that is also that we use the LC retention time, so what time it takes to pass this LC column. because this is related to properties, as I said. So there is a correlation between log P, the lipophilicity of a compound, and the retention time. So we have a prediction model based on textile chemicals. And if the software suggests a compound, we look at the log P, and then it cannot deviate more than ±2 minutes from this prediction. Then this suggestion will be discarded. Next slide, please. So if we are lucky, we can also confirm. So this is the final step of identification. If we can confirm with a standard compound, then we call it identification at the level 1, the highest confidence level. So this is just an example of nitrophenol detected in, it's a pesticide detected in a garment. And in this case, as you see, both the retention time, it has the same retention time, but also the same mass spectrum as a purchased standard compound that we have injected. Next slide, please. So by using a combination of non-target and suspect screening, we have been able to identify in more than 80 garments, we have been able to identify different problematic, I would say, compound classes. And these garments are bought on the Swedish market. They are mostly imported. And as you can see, the origin is mainly from Asia, or from all over the world. And also mostly synthetic or blended textiles of different colors. The main compound classes, we detect many more, but the main compound classes are benzothiazoles, benzotriazoles, quinolines, and quinoline is a possible, probable human carcinogen, and then you have derivatives of quinolines. And anilines is another worrying group because some of these anilines are considered as mutagens, but also skin sensitizers. And some of them occur at levels up to 1000 ppm. So that is a huge level. Then we have phthalates, as in all other types of materials, azo dyes, nitrophenols, acridine, organophosphate esters. The next slide, please. Of course, it's also important that we can, for a greener society, that we can recycle textiles and use recycled textiles. And our colleagues in the same department, MMK at Stockholm University, they are making new materials. So they enrich cellulose in nanocrystals from recycled clothes. And then, of course, it's meant that the cellulose is meant then to be spun into new fibers and for production of new textiles and clothing. And of course, it's important then to follow the the life cycle of chemicals in this, both in the materials, but also in the waste. Next slide, please. And one way to, of course, we can, as usual, we can quantify and we can identify many compounds, and also in the recycled textiles, regardless of laundry, etc. Of course, they have then decreased the levels in the materials, but then on the other hand, they have been released into the aqueous environment. Anyways, this is a way to compare starting material, the recycled garments, and the produced materials, in this case, the cellulose nanocrystals. So we have done the non-targeted and suspect screening of this. This diagram is called the volcano plot, and I need to explain it. On the x-axis, we have the difference in levels between starting materials and produced materials. So if it is 0, there is no difference between the levels in the materials. More to the right, it's more in the produced materials in the nanocrystals, and to the left, it's more in the starting material in the recycled garment. And then the red triangles are those that we have identified, and we have searched in the ECHA database. And the gray, you can see there are many features that we have not been able to identify. Anyways, we can see that most hazardous chemicals are actually decreased a lot in the produced materials. Those we find mostly to the left. And on the y-axis, this means the significance of the analysis. For instance, as an example, we can see quinoline here. We have much more in the starting material, in the recycled textile, compared to the produced material. And in general, we can see that the chemicals decrease, the most hazardous ones. But as I said, then they end up somewhere in the process, so in the waste. So we are also performing non-targeted screening of the waste in this process. Next slide, please. Some conclusions from, not from just this presentation, but from our work, is that in both new and recycled skin closed garments, there are numerous of hazardous chemicals that could actually be taken up by human skin. Because what we extract are chemicals mainly below molecular weight of 500 and log P between 1 and 4. And this is the range where chemicals can be absorbed by human skin. It's important to do this life cycle analysis, and non-targeted screening is an important tool in this, to use it in the textile production, both from new and recycled materials. And also something that is really important is that we need much better control of textiles, especially those imported to the European Union. As far as I understand, there is almost no control of the chemical content. Next slide, please. And I want to finish just by saying that we have a method that we think could be useful for the industry or for control labs to check for the most problematic compounds in textiles. And this is in a way a very simple method. We just take small pieces of the textiles, grind into powder, load sample tubes, so-called desorption tubes, with this material and heat it up. And this chemical vapor containing very often quinolines and those aromatic anilines, those most toxic chemicals in textiles, that vapor is automatically introduced in the GCMS, and we get the data as a chromatogram and also mass spectra. It's still under validation, but we think that this method could be simple enough for a control lab to use for textiles, clothing, and also other materials. Yes. So with that, next slide, please. With that, I want to thank my collaborators, Josefin Karlsson and Tim Åström, who are PhD students in my group, Janis Sadixis, who is a PhD in my group, Conny Östman, Professor Emeritus, and also thanks to our co-workers in the Cellulose Nanocrystal Project, Professor Ajay Matthew and PhD student Maria Caldas. Thanks a lot for listening.
Thank you, Ulrika. And if there are no burning questions now, Ragnhild, I'll just rush along to the final bit, which is tying the knot on hazard and risk screening and its relationship to LCA. And this is very descriptive, but essentially I just want to hammer home that, of course, you can either deal with hazards as they are and rank them and make them proportionally representative towards each other. But of course, a hazard only becomes a risk if you're exposed to chemicals. So the area of safe and sustainable by design is being achieved in various areas of chemical endeavor, actually, and there is a history book here, but essentially by performing iteratively reduction or ultimately elimination of hazards, which obviously is the mantra that we're seeking. or using, but also reduction in exposures, of course. Therefore, the risks are reduced, or both of these. Ideally, one wants to minimize the hazards or remove them, and at the same time work with reduction in unnecessary exposure, both of these. And my experience in this area comes from working in the pharmaceutical industry, and the process is very similar to the one we're designing in SafeChem here, that the safe and sustainable by design that was used in, or is used in, the pharmaceutical industry for many years uses what's called the make, test, and analyze, design, make, test, and analyze cycle. And these are small iterative cycles where chemicals are designed, made, tested, and analyzed, and properties are then modified basically on an iterative cyclic basis here in order to minimize the, in this case, the toxicity profile of the potential drug candidate, but optimizing, still allowing the pharmacological effect to occur, and obviously optimizing the properties versus the pharmacological properties versus safety is the reason for this iterative cyclic approach, using hazard assessment early on in design of process, actually. And so this is very similar to what many areas of chemical process endeavor are adopting at the moment. And this is something that's essentially a focal point in the new chemicals policies from the EU, that this should be approached from this particular angle. And so early hazard warning is obviously a very, very essential part. And this work package is essentially about hazard screening and also exposure screening. We've presented a tiered approach for hazard screening using in silico and in vitro toolboxes, and that has been allowed, or that's provided rapid comparative hazard assessment, and that's being applied in our various case studies within the program that we're working with at the moment. And also early application of targeted, non-targeted screening, as we've just heard from Ulrika, allows us to identify and potentially quantify suspect chemical exposures. Some we may know about, some we need to define because they're new. And these can be combined with the hazard assessments to yield the starts to risk assessment. And risk assessment can be taken at different levels, of course. We're not talking essentially directly risk assessment for regulatory submissions. We are talking about decision-making processes, but they are based on both hazard and risk and exposure. And so Mr. SafeChem, in our particular work package, we're focusing on the agility in these predictions. So it's this, again, back to being able to quickly apply the tools in the toolboxes for efficient decision making when you're adapting or developing a chemical process, which we have in our case studies. The data can be used and utilized fully later on in fuller risk assessments for regulatory purposes, because the tools are essentially recognized and run according to standard operating procedures and so on and so forth. They are the data is relevant there, but whether Mr. SafeChem in its two phases gets towards actual regulatory submissions on process development in terms of reach, then we'll have to see how that develops. But primarily agility on decision making when we're adapting process. And the data, importantly, are directly admissible into LCA and alternative approaches for both human and environmental impact. And just to quickly praise the life cycle assessment, of course, is important. It takes in the entire lifetime of the chemical and the process we're dealing with. And we have a work package in Mr. SafeChem, work package 5, where life cycle analysis is applied, where we're dealing with the full life cycle of the product and the raw materials, the acquisition through production, the use, end of life treatment, recycling, and final disposal. All of these are components in the life cycle assessment, and the data we generate from hazard and exposure assessment in work package 3 in our various case studies are directly admissible into life cycle analyses. And of course, the hazard screening and early warning and proactivity that we've just described, the ability to pick this up, is then provided into life cycle assessment and management. Data gaps might be identified and they can be iteratively filled to make fuller life cycle assessments, and these being applied to the design and management of the chemicals, the materials, and the processes in our case studies. And so this is the approach that we've adopted. And in the toolbox, the life cycle analysis toolbox, we have various entry points into the toolbox, but of course the toxicity profiling, the human and ecotoxicity profiling are essential from the footprint analysis that we do, essential inputs into the life cycle analysis toolbox. But there are various ways in which this data can be applied into the toolbox for life cycle assessment, which is something we may come back to in further breakfast seminars later on. So this is just a cursory description of how the data is then inserted into life cycle assessment analysis. So that's the end of the presentations. Now we'll just stop I'll just stop the sharing. Boop. There we go. And we'll go back to the questions that arise in the talk. Now I've got to try and get that up now. Where is that now?
I can read it to you first.
Yes, fine. Carry on then.
The first question is, was when Ulf had his presentation, what models do you use for the prediction of readily biodegradability, and how well do they perform in your experience? It was a question from Stephanie Lawrence.
Yes, what we use is a set of structures, about 1,000 of them, which are actually published by the National Institute of Technology and Evaluation and from Japan. They're all based on the OECD test guideline 301, which is activated sludge pre-cultured with synthetic sludge, basically either 14 or 28 days. And as far as we can see from our internal validations, of course, they seem to perform fairly well. Now, these are all classification as models, which stating that yes or no, degraded or not, according to these test guidelines, basically. I hope that answers the question.
Yeah, thank you, Ulf. Ragnhild, next one.
Yes, we have some questions then for Ulrika. Gregory Peters asked, you found these compounds in 80 plus garments. How many garments did you test in total?
Yeah, in total it is about 80, so it's more than 80. I would say between 80 and 100. Mm.
Okay, and
but we are continuing this study. So. Mm.
Then Åke Bergman asked, what about the availability of authentic standard reference compounds?
Of course, it is restricted, but we have, for instance, regarding azo dyes, I mean, there are several hundred types of azo dyes that can release, I mean, different dangerous amines, for instance, and there we have it's not easy to get them. So as pure compounds, most azo dyes are very dirty still, but bought as standard compounds, maybe we have, I don't know, the PhD students know, but I think it's about 20 standards only. So it's very restricted, the number of standards we have. And therefore, such work as predicting ionization efficiencies in electrospray, for instance, could be very useful, so we don't have to use standards for everything. Mm.
Then there's another question for you. Can you see a difference between new and washed clothes?
Yes, so we see that some chemicals, so we haven't investigated aromatic amines, it's not finished yet, our study, but after 1 or 2 laundry cycles, they are still there in the textiles. And for instance, so they are, it seems that they even can increase after laundry. And we think that could be because of thionite in washing powder that can degrade azo dyes into amines. But we have to investigate this further. And quinolines, and they stay, I mean, maybe 50% is still left after 5 laundry cycles. So it depends on what type of chemical it is. Some remain and some are released into the aqueous environment more.
Many questions for you, Ulrika, here. Anna Skog asked, what libraries do you use for identification, since you mentioned the 80% match? NIST and the match for REV or FOR? I don't know what this stands for, but you know better than me.
Yeah, NIST library and Mona. Yeah, so there are different libraries. Maybe my PhD students should correct me. They are more into the details of this. So Tim, maybe you are here, so maybe you can.
Yes, I'm here. But yeah, that's right. The NIST and the Mona library for sure. And also when we use Compound Discover, then we look in MSAT Cloud and MSAT Vault libraries. So that's right. There are multiple libraries that we are using when we are looking into the different compounds and trying to identify them.
Thank you, Tim.
Can I just add one thing here from a hazard perspective? One thing that Ulrika's group is working with us in Work Package 3, who are working with in silico tools, for instance, is that the data that she's generating, when she does identify things, then quickly either finding literature sources, if they're well-known CMR substances, for instance, obviously there's a basis for that. But if we have near liars that we really don't know very much about, obviously digitally moving those data directly into hazard assessment is actually what the pipeline is all about in Mr. SafeChem, and doing that from targeted or non-targeted screening. That is the challenge, is to get some sort of hazard assessment, and also use specific compounds when we know what they are and we're very well established. We can use them as beacon substances, what I call beacons, to when we change process, to really see if we reduce the production of particularly CMR. A number of those already Ulrika stated were known carcinogens, and they're well known. Obviously you want to monitor them and find out where they're going. And eventually you have to remediate those problems as well. That's another problem in your water flows. But that's the mantra, if you like, between the target and non-target data, and also the in silico tools that we're applying in this particular and other case studies. Just want to make that clear.
I have further questions, or is there anyone else? One question more for Ulrika. Well, not the last one, but this one is, what do we know about the persistence of these compounds?
That I'm not the perfect person to answer this, actually. I'm not a toxicologist or environmental chemist.
Maybe
someone
help out.
Yeah.
Maybe I can help Ulrika. This would be if there are no data out there on those particular substances that we're focused on, then clearly we'd search them. So that's literature data, right? So, but if there is no data, then of course the in silico tools on biodegradation, bioaccumulation, provide some estimate, if you like, of accumulatability in the environment, but that's an in silico prediction. Then we also have biodegradation tools being developed at IVL, where specific processes can be monitored under various environmental conditions. So that's part and parcel also of the Work Package 3. We have that capability to actually do experiments on biodegradability as well, if it's an unknown compound or mixture in that case.
But also those aromatic amines, for instance, they are very reactive. So I wouldn't say they are persistent. They are more reactive and more problematic for humans, for instance. And then they are probably transformed to something else in the aqueous environment.
Absolutely.
Then we have more of a comment than a question. Lisa Filmreiter, you wrote in the chat, I want to argue that the Swedish fashion and textile importers are in general very diligent in managing chem contact in the products. We should worry much more about parallel imported items. More of a comment. Then we have a question from Peter Skagelind. Do we have data showing good and bad textile, and do we know why the good are good? This could maybe be used as a driver for new fashion companies to come up with a sustainable profile.
Yeah, what we can see is that eco-labeled have less of chemicals, but we are surprised sometimes to find also very toxic chemicals in those, even in baby clothing made from cotton. But in general, I would say also that cotton contains less of those aromatic amines and those more toxic compounds. But on the other hand, I mean, cotton has other problems like water consumption, and so it's very difficult for me to judge.
Obviously, that would be a very important component in the life cycle assessment of process change. You know, there are multiple. It's not just the hazardous chemicals; it's the carbon footprint, it's the use of water, energy, which also become components in deciding how to trim a process, as well as the hazardous emissions as well, primarily, of course.
The next question might be for Ian. It's which cells are used in the cell painting assay? Is that your part or is it Ulrika's? I'm not sure.
That's my part, and that's Oscar, who, if he may be listening from SciLife SU, he has various models, and they're very basic. They're nothing fancy, I don't think. He paints probably the neuronal cells, for instance. He's probably also got a group of fibroblastic cells. I don't know the details. Oscar from Stockholm's University would have the answers to that one in his shop. But I think they're very associated with the kinds of high content biology that I showed you. So the neuronal cells will probably be there. Is Oscar on the line?
I'm not sure.
No, I don't think he is. Anyway, that question we can take away and answer directly after the seminar's finished.
And then the last question in the chat right now, anyway, is from Anna Steneholm. She writes, the 4-week cycle seemed to be a good approach in pharma, but what do we do about current substances like diclofenac that now seem to be available in the environment and that seem to often drive the need for MIF in the discussion on combined exposure to chemicals?
Oh, well, diclofenac's a legacy compound. It was designed and built many years ago, and it wasn't built with, if you like, green and sustainable process in mind. So it's one of the legacy compounds. Today, I would say that the pharma industry, the DMTA cycle is very much shorter. I would say it's a matter of days to a week these days. And that's a fairly old picture. And much of it is done in silico, actually, today. But they do build in also the biodegradability aspect of the drug. So green chemistry is also built into that process as much as they can. Obviously, they're interested in a pharmacologically active compound with low toxicity profile. And then on top of that, they're interested in the fact that it will not bioaccumulate. So diclofenac's a problem. It's a legacy compound built in the old way. I agree that that's a problem. I have no better answer than that, I'm afraid. Was it Anna? Yep.
Yes, and that closes the questions in the chat, but I think we have some, if we don't have any more questions from anyone.
So raise your hand now if you've got a question. So if there's anybody who requires any more information from the use of our toolboxes in our case studies, specific case studies, and particularly how the data is generated and utilized in an interface towards life cycle analysis, please contact me, and particularly from the LCA perspective, Anna Holmqvist and Anna-Karin Hellström will take care of that from our side. But any questions around our toolboxes and utilization, please address them to me, and I'll make sure you get an answer.
Then just Ian, if you could put up the last slide, because we have more webinars coming up in February.
I'll do that.
You can share your screen. We have a plan of all the work packages to have webinars, and now this is work package 3. And the 8th of February, we will have work package 4, which work with design and management of chemicals, materials, and processes. So we have not yet made the invitation, but it will come shortly. And I hope you will get it in your email, or otherwise you will find it on our website later on after the new year, I would say. I hope you will join then and get some more information on the work in the Mr. SafeChem program. So that's it from me.
Well, thank you very much, everybody, for being part of the seminar, the webinar. We hope you've had an interesting time and maybe learned a little bit about the inner workings of this rather large platform, this part of the motor in Mr. SafeChem. And again, if there are any questions around it, further questions, address them to me and I'll make sure you get an answer. And so exactly 9:15, I'd like to say thank you for us on behalf of us, and hope you have a good day the rest of the day.